Okay. So we have been talking about making genomic DNA libraries and then or Cdna libraries as the case may be. I think I've reiterated again and again that when it comes to genomic libraries, generally, prokaryotes, by virtue of the fact that they don't have any introns, genomic DNA library serves a very useful function, right? Nobody would go and do a Cdna library for prokaryotes. On the other hand, when you come to eukaryotes, because they carry a lot of introns, right? So you need to think whether you want to make a genomic DNA library or a cDNA library, right? Especially given the fact that if you were to consider human genome, literally 98% of the genome, okay, codes for literally nothing, right? 98% of the genome codes for nothing. These are all structural elements. That means they are promoters, enhancers, so many other varieties of these controlling elements that are present. And your gene coding region is only 2%, right? So if you are looking for a novel gene, or suppose you're looking for a gene homologue, right? Then you would probably be better off trying to use a cDNA rather than a genomic DNA. But all said and done, you may still use genomic DNA for eukaryotes if you want to understand, for example, you have cloned a gene and you want to understand how this gene expression is controlled by the promoter, what are all the upstream elements that are present in this promoter. Then you may have to have a genomic library, right? And we also saw some mathematics behind, how many clones you would need to screen in order to find a decent probability that you are going to end up getting this gene of your choice. It's like, genomic DNA or cDNA, it doesn't matter. You're going to generate lots of clones. The idea is you need to you're literally looking at a needle in the haystack. I mean, you have a big haystack and you have a needle there. You don't know where the needle is stuck, and so you need to screen that needle, right? What could you do? Suppose literally, suppose there is a needle in the haystack. What can you do to pull out that needle? Simply pass a magnet through it. Hopefully the magnet will attract the needle, and you could basically discard the entire haystack, or you could remove that needle from the haystack very easily, right? So essentially we are going to do a screening, and the last time we saw from that lecture at MIT, that professor explaining beautifully, right? How do we screen for these clones, right? Clones are screened by using complementary mutations, right? You would use certain mutants where you could have functions being gained. You could also use different ways by which you could screen. Your gene could be coding for a protein, which means you could do a functional assay. Okay. Suppose functional assays are tough, then you could probably raise an antibody and you could screen, using immunological techniques. So screening can form, many different things. But in today's class, we will just go back and revisit, how do we make genomic libraries? Okay. So there are two things that we can do. The easiest thing to do is to cut your genomic DNA and clone them into plasmid vectors, right? Plasmid vectors are ubiquitous. You can probably walk into any molecular biology lab, they will have plasmid of your choice. In all probability, people will use plasmid called PUC 18 or 19, which we talked in the cloning vector part, right? So PUC 18 or PUC 19 is commonly used as a cloning vector. PUC, P stands for small p stands for plasmid, UC stands for University of California. I mean, this was done long time ago. So the name stuck. Okay. So PUC plasmids are very, very common, and you know those are the vectors. Okay. As we saw in the class, you know, plasmid can carry, theoretically, it can carry up to 20 kilobase pairs. But unfortunately, those theoretical considerations, you know, don't seem to work. And many a times when plasmids carry a big chunk of DNA, they tend to get lost during subsequent growth in E. coli. So they tend to get rearranged, you know, many different kinds of things happen. So what has been found practically, right? So there is a theoretical consideration and there is a practical consideration. Practically, what has been found is that about 2 to 4 kilobase pair is the maximum that you can use in E. coli plasmids, right? If you're trying to use PUC 18 or PUC 19, for that matter, many of the other plasmids, anywhere between 2 to 4 kilobase pairs is what is the best this thing, you know, best optimal size of your genomic DNA, and that translates literally thousands of clones if you were to make a genomic library in E. coli. Okay, people have used very simple phage, lambda phage. I'm sure you must be aware of lambda phage during your you know, senior school days. Lambda phage is a phage that has both lytic and lysogenic life cycles, right? So it turns out that within the lambda phage, about 20 kilobase pairs of its genomic DNA can be removed, and the phage behaves as though nothing has happened, right? What has happened in those 20 kilobase pairs is that all the elements that are needed for lysogenic life cycle are there. So you can remove it, and the phage essentially you know, multiplies in E. coli by lytic cycle. Okay, so those 20 kilobase pairs can be removed, and you can put your genomic DNA into that. Okay, so now you need to screen much less colonies. Okay, nevertheless you still need to screen, right? So people have evolved different you know, techniques by which this can be done. In today's class, we will finish what we started last time, right? How do we make genomic libraries in the first place? And then we will you know, reinforce, you know, what we saw in that MIT professor, you know, we will reinforce that in our subsequent classes. Okay. Professor Eric Lander, that's his name. Okay, is now the scientific advisor to the current president. All right, I'm trying to share my screen. Hopefully, this should be. So this is where you know, the question is, if you want to use a restriction enzyme, what would you use? As I said, some of this is you know, historical. So we're going to look at a historical account of how people did genomic libraries, right?
Consideration that is very important to remember is that
Is
we
the
create
screen visible?
genomic libraries so that we can sequence them
Yes,
and
sir.
Audible?
Yes, sir.
Yes,
No.
sir.
look at the entire genomic sequence of that organism or the sequence of the chromosomes that are in that organism. So in order to do that, because you know, restriction enzymes will cut the genome in many fragments, you want to have the clones in such a way that there is an overlap between the clones, right? So ideally, if you can get one clone to represent one gene, that is the best case scenario. But we know that eukaryotic genes are very, very big. You also, the restriction enzymes will cut indiscriminately between the gene and in anywhere. So you will at least try to achieve to have a specific genomic region to be represented by at least a few overlapping clones so that you can reconstruct that entire genome. So a 4-base pair cutter enzyme is generally preferred, and one of the commonly used enzyme is Sol3A. And the reason for choosing a 4-base pair cutter is as follows. that we know that the frequency of a 4-base pair cutter site to be found is every 256 bases, assuming the equal distribution of nucleotides. Now, this DNA fragment here shows it's a human DNA fragment, and these are all the SAW3A sites that are present. Now, the scale is given here, 2 kb. Now, you can see within 2 kb, there can be multiple sites, but they are not exactly evenly distributed, right? And that is because of the bias, codon bias of the genome, which may be more GC rich. So if we let SAW3A to cut this genomic fragment completely, then there'll be many small genomic fragments, and it will be very difficult to reconstruct the sequence of this entire fragment. So a trick that is used by researchers to tackle this problem is to carry out what is known as a partial digestion. And this partial digestion is nothing but you dilute the restriction enzymes quite a bit and then add very little of that enzyme, and then you withdraw the samples and keep checking to obtain the fragment size which are desirable. So for example, if you want to generate about 20 kb fragments, then you have to basically set up the digestion reaction and standardize as to how much SAW3A and how much time is required to digest a particular genome into about 20 kb fragments. You can check this on the agarose gel. So what happens during a partial digestion is that not every site will be digested by SAW3A. So it will be making random cuts at different positions. And because of that random cuts, you will obtain overlapping fragments. So in a cartoon, it is shown here. So supposing this site is digested, then maybe this site is digested, this site is digested, this site is digested, and rest and this. So you generate all these different fragments. And because they are overlapping with each other, when you sequence all these fragments, by aligning all of them together, you can get a sequence of this human DNA from this end to this end. So this is what researchers try to achieve when they are creating a genome sequence. Now, in reality, the actual number of clones that are needed to be very certain of finding a particular sequence is much higher than the minimum numbers that I talked about before. That, okay, for a human genome, if you have 750,000 clones, then it represents the entire genome. So why the actual number is much higher? The actual number of clones is much higher because, as you saw in the previous slide, the cuts that are made are random. Now, when you are screening the colonies, how do you make sure that you will find, say, for example, this particular fragment, right? You can only find it in a random, so you cannot possibly check all the colonies that are present in the genome library one by one. And so you want to spread out your genome library on plates, and then you want to, so maybe you have 10 or 15 such plates, and you want to find the gene of your interest. So because it's a random choice, therefore, the actual number of clones need to be much higher to increase the probability of finding a particular gene of interest. So people have done this exercise theory.
Something that we discussed in the beginning, right? We had given some mathematical number that if you were to clone E. coli genome, how many clones would be needed, right? And since the probability is very low to find a single gene, in order to increase the probability, you will probably end up screening several times more than the actual number of clones. The actual number of clones was around 40,000, but we will instead screen probably 2 to 3 times higher, so that the probability of us hitting, or, detecting our gene of interest becomes much higher.
Critically, and they have come up with a formula which basically tells you what are the number of clones which are required. This is shown by n, so that you can find a particular sequence with a certain probability, which is p. So n is equal to natural log of 1 minus p, the probability with which you want to find the sequence. So for example, you want to be 90% sure that when you screen the library, you get the clone of your interest, then p becomes 0.9. You want to be 99% sure, p becomes 0.99. So natural log of 1 minus p divided by natural log of 1 minus f. So what is this f? F is nothing but the fractional proportion of the genome, as I calculated before. And this is basically the size of the fragment divided by the genome size. And this number you substitute in the fraction proportional of the genome. So now let's see what are the number of clones that are required and how it changes with the probability with which you want to find the sequence and the fractional proportion of the genome. So in other words, how do you choose the fragment length and what consequence that fragment length has on the...
Pay close attention to the explanation in this slide. Okay, this is a beautiful summary of how will you screen a library and how many colonies need to be screened.
Number of clones that are required. Okay, so let's take a genome, which is human genome. So 3 into 10 power of 9 base pairs. And we consider two different fragment lengths, 4096 base pair and 20 kb fragments, so 20 into 10 power of 3 base pairs. Let's put p at 99%. So you want to be 99% sure of finding any sequence that you're looking for in the gene library. And so the fractional length will be, in this case, 4096 divided by the genome length, 3 into 10 power of 9. And in this case, the fragment length is 20,000. So F will be 20 into 10 power of 3 divided by genome length, 3 into 10 power of 9, which is 0.6666 into 10 power of minus 5. Now you put these values in the formula. N is equal to natural log of 1 minus p divided by natural log of 1 minus f. So when you do that and do the calculation for these two cases, so if you have a short fragment length of about 4 kb, then you need to screen 3.3 into 10 power of 6 colonies of a human genomic DNA library in order to be 99% sure of finding a gene of your interest. As you increase the fragment length to 20 kb, now the number of clones comes down marginally. It becomes 6.9 into 10 power of 5, which is definitely a lot less than 10 power of 6, tenfold less. So because of these considerations, vectors that can take larger inserts are preferred so that the number of clones that are required to be screened is much lower. And because of that, instead of using plasmid DNA libraries, people prefer to make genomic DNA libraries into either lambda phages.
So are the reasons clear why, although it's easier to make a library in plasmid, people don't use plasmid libraries, Instead, they use either a lambda phage or a P1 artificial chromosome or any of the other artificial chromosome based approaches. Of course, it also depends on the genome that you're talking about. Suppose if it's a small genome, then it probably doesn't matter. But for something like mammalian genome, you may need to have other alternates. It's virtually impossible to screen that many number of clones in a plasmid. Okay.
Based vector. which can take anywhere between 10 to 40 kb fragments, depending on the kind of lambda vector that you're using. And so, for example, if you're making E. coli genomic DNA library, you can probably manage with that. But with increasing genome size, maybe you need better cloning vectors in order to reduce the number of clones that you need to screen. So you can use a P1 bacteriophage-based vector, which is P1 artificial chromosome or PAC vector. It can take anywhere between 100 to 150 kb fragments, or you can use bacterial artificial chromosomes, which will take about 300 kb fragments, or yeast artificial chromosomes. We have talked about it before, which can take up to anywhere between 1,000 kb or even more fragment that can be cloned into these vectors. So these vectors are preferred to make the genomic DNA libraries. And as the genome size increases, then you want to use a vector that can take much larger fragments. So let me just remind you about the lambda vectors. And you remember there were two types of vectors, lambda insertional vectors and lambda replacement vectors. And while the lambda insertional vectors can take up to 10 kb fragment, the lambda replacement vectors can take up to 40 kb fragments. So essentially both the vectors, when you clone the gene into them, they result in the inactivation of CI gene. And this CI gene, if you remember, was important for lysogeny. So when you disrupt the CI gene due to cloning, then the recombinant vectors will basically undergo the lytic cycle. So when you, after cloning, you infect the E. coli host, and so both non-recombinant and recombinant colonies, bacteriophages will grow. And so the wild type will mostly be lysogenic, whereas recombinants will always be lytic. So you get a cloudy plaque where it is the non-recombinant bacteriophage vector, and the clear plaques because of the recombined bacteriophage vector. And a special strain like HFL strain of E. coli is used, which basically stabilizes the CI gene. So all non-recombinants will not produce a plaque because they are lysogenic. And all the recombinants, because they don't have the CI gene product, they will all produce lytic colonies, and therefore they can generate the progeny. So you will have clear areas on the lawn of E. coli cells, and those plaques, because they have been generated by lysing the E. coli host cells when there are enough number of bacteriophage particles that are produced. So those clear areas or those plaques will contain these bacteriophages, which contain the recombinant DNA. So with this background, now let's talk about what are the various ways in which the gene libraries can be screened. So there are three different basic approaches. One is the hybridization analysis, second is the PCR based analysis, and third is the immunological technique, which is used for only cDNA expression libraries and not for the genomic DNA libraries. So let's first talk about the hybridization analysis. So in this case, you have a master plate where you have plated out your recombinant E. coli on the agar plates. In reality, you don't have only a single plate because there are large number of clones that are there that will represent the entire genome. And so if you put them all in one plate, all you will get is a lawn. So you want to spread it out into different large number of plates so that you can get well separated plaques. Now you take another.
So as we saw in the previous calculation, assuming that, every 4 kilobase pair there is a cut, you are supposed to screen about 3.3 million colonies to get one single clone, right? If it is 3.3 million clones to be screened, how many Petri plates would that take? It would take a large number of Petri plates, right? And typically, each Petri dish the normal Petri dish that you see here in the lab can have up to about 100, well separated colonies, not more than that. Beyond that, they are so crowded that it becomes very difficult to pick out each individual colonies. About 100 colonies, 100 colonies randomly distributed, evenly spaced, it'll be about 100 colonies. So if it is 100 colonies per plate, then you can imagine if it is 3.3 million clones to be screened, how many Petri dishes will we be using? So that's a huge number, right? That is, but then, as I said, it also depends on the genome, whether you're screening a mammalian genome, are you screening a smaller genome, it depends on this thing. But this example is talking about a smaller genome where you're probably going to have about, say, 20 or 30 Petri dishes, right? Not more than that, okay? We will not worry about, for the time being, those n number of plates.
Nitrocellulose membrane. So I will just show the cartoon for one single plate, but essentially you have a collection of plates on which this analysis will be done. Excuse me. So you take a nitrocellulose membrane and you gently put it on top of the plate and you lift it very gently so that the plaques, they do not get dragged and mixed with each other. And you also want to mark the orientation of the way this nitrocellulose has been put. Normally people just make a couple of punches in 2-3 different places so that they can align it later on with the master plate. So you lift the nitrocellulose, and now what is going to happen is all this plaque material is also transferred onto the nitrocellulose membrane. So you reserve this master plate, you keep that, you store that properly, because then you will need to pick this plaque depending on which one of these are actually the clone, represent the plaque of your interest. So now, because the plaques have been transferred and they will contain the bacteriophage vectors containing the DNA of interest, right? So each of them has a different DNA fragment that is there. So you lyse them, denature the DNA, and hybridize to a DNA probe. And if this DNA probe is labeled with radioactivity, you do autoradiography. Nowadays you can also use a fluorescently labeled probe, and you develop it on a film, photographic film, or a phosphorimager. Then the probe will hybridize to whichever plaque contains the gene of interest. So supposing one plaque lights up on your autoradiograph, so this will be one signal, and most likely this is the one. So now you align this nitrocellulose with the original master plate, and you know this is the plaque that contains the signal. So you go ahead and you pick this plaque from the master plate, which will contain the bacteriophage with the desired DNA fragment. You isolate the DNA and then you identify the gene by sequencing. Now, similar kind of analysis can also be done for bacterial colonies. And that is done by what is known as replica plating, where you have a velvet covered head. It's like a rod that you gently press on the master plate and then plate it on several different plates and grow them in order to have a replica of all the colonies that are present on the plate. And also you make a replica on the nitrocellulose. Now, instead of plaques, these will represent the E. coli colonies. And so you lyse the bacteria, denature, hybridize to a probe in a similar way that you've done before. And instead of picking a plaque, now you align and pick up the E. coli colony from one of the replica plates that you have created from the master plate, isolate the plasmid DNA and sequence. Now, if you're doing plasmid cloning, then there will be a large number of E. coli colonies. And so the replica plating does not give a very clean results because the E. coli colonies, they give a background when you develop the film. So picking up what is positive, what is not positive is a little difficult. Secondly, even though you may lift very carefully the colonies, there is a lot of smudging and mixing of the colonies because E. coli colonies are very, very small. But that can be done if you have very few colonies and they are very well separated on your master plate, then replica plating can work very well. So essentially what you have to understand is that you can hybridize whatever, whether it's a plaque, whether it is E. coli containing a plasmid or a yak or a BAC containing the DNA fragment of your interest to a DNA probe. As long as you can do that, you can pick up the original clone or original plaque that contain the gene of your interest. So these probes, they are of two types, the homologous probes or heterologous probes. Homologous probes, as the name suggests, they are from the same organism. And these are the DNA fragments which are the exact complement of the sequence that is being searched. So for example, you have a partial fragment of a gene. Say you have a mouse gene, you have a partial fragment of that gene, and now you want to pull out the entire gene. So you make a probe out of that fragment that you have and screen a genomic DNA library, and you can pull out the entire gene. This is also done to identify the genomic location of different genes. Also, if you have the genomic library from normal and diseased tissue, then you can use the probe to pull out those genes from the normal and diseased tissue, sequence them to study what type of mutations might be there in the disease condition. The heterologous probe is a similar to the gene being sequenced, to being searched, but not totally identical. And this is generally done to search for homologs of genes in different species. So for example, you may have a gene sequence, a gene fragment from a yeast gene, and you want to pull out the homolog of that gene from, say, a mouse cell. So you prepare the probe of the yeast gene fragment, and then you search a mouse genomic DNA library and pull out the gene which is homologous to the yeast gene in this manner. So these are the different kinds of probes that can be used. And of course, the probe has to be labeled so that it gives a signal and you can pick those signals. One thing I mentioned forgot to mention in the last slide, in general, it is not that you get only a single positive plaque or single positive colony. In reality, you get multiple colonies or multiple plaques because you remember that the genome has been fragmented and there are many overlapping fragments. So you may get several of them, and then you may need to repeat with a slightly different probe in case you feel that some part of the genome is missing. Okay, so these probes, they are usually labeled. So one can use 32P labeled probes or fluorescent probes. Nowadays, there are companies which can make fluorescent probes for you at a cost. But whichever way they are labeled, you can use a number of techniques, a number of tricks to label them. You can either use nick translation, so that the probe is labeled at multiple places, most likely. You can also do an end labeling of the probe, so that, for example, 5' end of the probe contains the label. You can also use random priming and PCR method in order to generate a probe which is The other method of screening is the PCR base screening, and this is much more labor intensive because it works on the principle of dilution method for colony identification. So here all the colonies, they need to be transferred to these 96 well or 384 well plates. Of course, it can be automated, so one can automate these steps also. So in essence, what you do that after transformation of the E. coli, the colonies are picked and you can pool certain colonies in each of these wells. So for example, you may decide to pool 50 colonies per well and then you plate it in a 96 well plate. Then you isolate the DNA from all these wells and then you do a PCR using gene-specific primers. Now, because of the overlapping nature and because the same fragment may be present in different overlapping clones, you may get a number of different clones which light up upon, which basically are positive in your PCR reaction. So you pick these positive wells, and because initially you had 50 colonies in each of these wells, now you dilute each of these. So supposing there were 50 colonies, and if you dilute it, many times in such a way that now when you plate them again, it is 1 colony per well. And then again you do the PCR, okay? You may again hit certain number of colonies which are positive. And so you can repeat this kind of test, dilution PCR several times till you are very sure that you have the PCR coming up from a single colonies in each of these wells. Then you sequence them and recreate the entire genomic sequence of that region. So this PCR-based screening can also be automated and done much more quickly. So to summarize, this part of this lecture is that I talked about the gene libraries, which is nothing but a collection of DNA fragments which represent the entire genome of an organism. And these fragments of the genomic DNA can be produced either by shearing of the DNA, say by brief sonication, or partial digestion by a restriction enzyme. And in general, a 4 base pair cutter is used so that many overlapping fragments are generated. And cloning a vector could be either a plasmid, which is not preferred because you generate too many clones that need to be sequenced and screened. But for larger genomes, bacteriophage lambda-based vectors or BAC vectors, PAC vectors, or YAC vectors are used. And of course, the choice of the cloning vector will depend on the genome size. The screening of the genomic DNA libraries can be done either by DNA or RNA hybridization or by PCR-based methods. And genomic DNA libraries are very, very useful for genome mapping and sequencing of the genomes, for finding novel genes. For example, sometimes, in many cancers, the genes which are not normally expressed can be expressed. Another complication with cancer is that not all genes are expressed in all the tissues. So when you're dealing with a particular type of cancer, then the library prepared from that particular tissue is more informative compared to a library prepared from any other random tissue. So genomic libraries, they are important for finding novel genes and also for finding homologous genes, and also they are very useful for finding the drug targets. So for this part of the lecture, you can refer to these 2 books, Molecular Cell Biology.
All right. So these are the references. In fact, I had mailed all of you a book in the beginning. right? That book also gives in lots of details, all these processes. I'm sure, if you go through it and you find any difficulty, we can discuss that in our subsequent classes. Okay. So please go back and refer to that book. Plus, I think there are additional resources that are available from NCBI database. Okay. So you can, these books are available from NCBI bookshelf. Okay, so you can check it out. It's given in a very simple, easy to understand language. Okay, so this was all about genomic libraries, right? As I keep saying, genomic libraries have a lot of utility, right? Especially when you are trying to understand gene structure, when you want to understand, what this piece of fragment, what this gene, what this DNA sequence is coding for, right? It's very useful. But that is good for prokaryotes, probably not so good for eukaryotes, right? Especially the mammalian genome, right? So there we will be looking at the cDNA libraries. So when it comes to cDNA libraries, the beginning part is a little different. In genomic library, we are basically taking genomic DNA and subjecting them to restriction enzyme digestion, either with a 4-base cutter or a 6-base cutter, and then we are going to clone them either into a plasmid or into a phage, lambda phage, or one of those things, right? Bacterial artificial chromosome or yeast artificial chromosome, depending on the genome size. Suppose somebody gives you, say 50 kilo base pair genome, which is lambda genome. Nobody would ever go and try to do this, in with P1, right? They would rather, given the fact that you can clone up to anywhere between 2 to 4 kilo base pairs, if you were to screen a plasmid, plasmid library would be more than enough, right? Randomly, if you were to screen, assuming that your plasmid is carrying 4 kilo base pairs, 10 clones will give you 40 kilo base pair coverage, and probably having about 15 clones will give you what is called as 1x coverage. That means you can you can have the entire genome represented once, right? Theoretically. But for general purpose, for screening, people generally do 2 to 3 times more. And so you end up probably screening a little more, maybe about 50 clones, and you are going to have what is called as 5x coverage, 5 times the coverage than what is actually needed theoretically. Okay. So it's good. So depending on the genome size, you decide whether you want to clone your genomic DNA in the plasmid vector, or is it going to be lambda pages, which we will do it in subsequent classes. Okay. Now, for a cDNA library, as I said, cDNA libraries are very useful when you want to understand the genes in eukaryotes, and your starting material is going to be messenger RNA, right? All eukaryotic messenger RNAs have a poly A tail, so you can actually use very smart things like use a series of T vectors, T oligos, or primers that have a series of T's in them. All these T's will go and bind to the messenger RNA, right? And from there, you can use an enzyme called reverse transcriptase, which will convert the information that is contained in the RNA into the form of a DNA base, right? So reverse transcriptase is a RNA dependent DNA polymerase. So it can convert the information that is contained in RNA into a DNA, right? So you can convert that. So that results in the formation of first strand. Okay. Now the first strand by itself, is not very useful because you can't clone a single stranded DNA. So somehow you need to make this first strand, which is the copied DNA, cDNA, into a double-stranded fragment. Okay, but you also realize that you have RNA sticking to it, right? RNA is complementary, so RNA is going to stick to it. So you use an enzyme called RNase, right? So RNase will come, it will chew up it will cut all the RNA into small fragments, right? And so now you have single-stranded cDNA. Okay. Now you hybridize this to certain primers. These are called as random primers. They have random sequences. Okay. So the probability of them going and finding a complementary region in the first strand, single-stranded cDNA is very high. Then you would use an enzyme, right? DNA polymerase that will polymerize all these short oligonucleotides and will create the second strand. Okay, now the first strand and the second strand are now perfectly complementary to each other. Then you will use, either an adapter that we discussed in the, a couple of lectures ago. Adapters or linkers can be used, and they can be cloned into appropriate cloning vectors. Of course, these days, it's easier to PCR out your gene from a pool and then go ahead and proceed for cloning. So, some of these, as I said, are historical techniques. These were used historically, right? People nowadays have the ability to sequence anything and everything can be sequenced right now in today's technology, right? So people would much rather go and sequence the entire genome. Okay, although it may take time, but once you have sequenced the entire genome, right, it becomes a repository, right? It becomes a database, and from the database you can very easily figure out, where the promoters are, where the beginning of the gene is, how does the gene end, right? And all those other sequences can be very easily assembled in vitro. So you can actually have a basic bioinformatic tool, and you can assemble the genomes very easily, and you will be able to simply do an analysis. Once you have the raw sequence, all you need to do is go back and do simple blast with your or homology search. You will be able to figure out whether your genome that you have sequenced, does it contain a gene that corresponds to E. coli, that is found in E. coli, or say something that is found, say you are looking at a virulent pathogen. You can see whether that has a gene that is associated with virulence or not. The reason I'm telling this is because if you look at E. coli, right? E. coli is a normal, non-pathogenic bacteria, right? E. coli is found literally in, everywhere, right? E. coli is there everywhere. But some of these E. coli can actually become pathogens, right? And so, you can go back and check whether this piece of DNA. So if you were to compare the genome of a normal E. coli and a pathogenic E. coli, you see a lot of differences in the pathogenic E. coli, right? So if you want to compare the genomes of two different E. coli, all you need to do is, if you already have, if you have sequenced, and if the sequence data is available, you can simply compare, whether these virulence genes are there or not. If virulent genes are there, then it is going to be a pathogenic bacteria. If it is missing segments of this virulent gene, then it is probably non-pathogenic. But in all probability, right, we treat even non-pathogenic as, probably pathogenic, because we need to be very, very careful when we are dealing with some of these bacterial species, right? So we take extreme precaution. We presume that it is pathogenic and deal with it accordingly. So so now it is the role of bioinformatics to take over, do all the analysis. You can do many different kinds of very interesting analysis, right? Suppose, as I keep saying, suppose this fifth semester batch went out for a remote place, found some novel bacteria, and you sequence the entire genome of this bacteria, now you can go back and check how many genes are common between this presumed newly discovered bacteria versus what is known, say, about E. coli or Bacillus or any of those other bacteria. What are the novel genes, right? And what these genes could be coding for, right? What are these proteins that have come out of these? You can simply do, comparison between that protein, the unknown ones, and you can search in the database. Does the database, NCBI database, does it have a similar gene? And if it is, if you see a similar gene, what is the function of that, and is this involved in doing a similar function in your bacteria? So many interesting questions can be answered. Okay, so maybe in tomorrow's class we will look at in Friday's class we will look at how do we make a cDNA library, and then the utility of these genomic and cDNA libraries. Okay, so any questions so far?
No, sir.
Okay.
Awesome.
All right. So, if there are no, so there will be a lot of overlap between this course and the FRDT from Dr. Naveen. So, I'm hoping that you guys are benefiting, right, from both the courses. Right? If there are any questions, you can mail me or WhatsApp me, and we can discuss it further. All right. So, see you on then Friday, right?
Yes, sir.
Okay, thank you.